Nitrosamine impurities are among the biggest quality concerns in pharmaceuticals. Learn how they form, why they pose serious health risks, and how manufacturers control them through scientific risk assessment. Even trace levels of nitrosamines can increase cancer risk over time. Understanding their sources, controlling contaminant, and following regulatory limits are essential for. Want to understand nitrosamine impurities in detail? Watch the full video to learn their formation, risk assessment, regulatory expectation, effective control strategies. Don't forget to like, subscribe, and share.
Look closely at a seemingly stable drug supply chain. A pill can pass standard tests for related substances, yet hiding inside that pristine formulation could be microscopic, part per million impurities actively threatening the entire batch. These trace contaminants are N-nitrosamines, including compounds like NDMA and NDEA. Under the ICH M7 guidelines, global regulators classify them as the cohort of concern, a highly potent group of mutagenic carcinogens. As a manufacturer, this places you in a difficult position. Regulators demand near-zero risk, but your reality involves tracking down trace impurities without grinding active production lines to a halt. You have a strict operational trade-off to navigate. You will have to sacrifice significant laboratory capital and analytical time to prove your product is clean. In exchange, you secure your market survival and protect patient safety. The threat of nitrosamines comes down to their specific biological mechanism. If ingested, they do not damage cells immediately. They are procarcinogens, meaning they wait for the human body to activate them. When they reach the liver, CYP450 enzymes metabolize the procarcinogen. This process strips the molecule down into a highly reactive electrophile, known as a methyl diazonium ion. This diagram illustrates the sequence. The top section shows conversion from the initial nitrosamine into the reactive methyl diazonium ion. Looking lower, these reactive species alkylate human DNA, forming structures like the O6-methylguanine adduct. This damage leads directly to cell mutation and increased cancer risk. Because of this severe biological potency, the FDA sets acceptable intake limits incredibly low. For many drugs, total daily exposure is capped at just 26.5 nanograms. That limit forces quality assurance teams into a corner. To find nanograms of an impurity inside milligrams of an active pharmaceutical ingredient, your lab has to push the extreme boundaries of modern analytical chemistry. To manage this, global health authorities enforce a strict three-step mitigation strategy for all approved and pending drug applications. This FDA flowchart maps out Step 1, a comprehensive risk assessment. You must evaluate the entire formulation matrix, APIs, excipients, and packaging. The goal at this decision node is identifying any chemical pathway capable of forming a nitrosamine, like a secondary amine reacting with trace nitrites. Executing this requires serious operational inputs. Teams have to collaborate across departments, pull years of historical batch data, and conduct exhaustive audits of external supply chains. This paper-based auditing delays immediate production schedules. However, completing it is a mandatory requirement to keep your drug application viable. A rigorous risk assessment is the cheapest defense available. But there is a catch. If this paperwork uncovers any theoretical risk pathway, you instantly trigger the need for physical, confirmatory laboratory testing. When you enter the confirmatory testing phase, the most accessible option is setting up a limit test procedure. This involves deploying standard analytical instruments, like an HPLC UV, to establish a binary safety threshold. The lab tests the batch, and it either passes or fails based on that preset limit. The operational advantages are clear. Limit testing gives you a rapid, cost-effective routine for releasing batches without requiring elite instrumentation or highly specialized technicians. The trade-off is resolution. A limit test lacks analytical specificity. It cannot tell you exactly how much nitrosamine is present. Because you are blind to the exact margin of failure, a positive limit test result forces an immediate escalation. You still have to pay for complex, quantitative methods to figure out exactly what went wrong. Limit tests work perfectly as an affordable first line of defense for stable, low-risk legacy drugs. But if you are trying to clear a complex formulation or resolve an ambiguous finding, they simply do not provide enough data. When limit testing falls short or the initial risk is too high, you must escalate to quantitative procedures. This tier relies on high-end mass spectrometry, specifically LC-MSMS or GC-MSMS, paired with internal isotopic standards. This table outlines the expected analytical specificity. The lab must configure the machine to monitor exact mass-to-charge ratios for multiple nitrosamine variants simultaneously. This equipment also requires painstaking sample preparation with specific diluents. Otherwise, the test could degrade the API and generate a false positive. This rigorous process delivers results. It provides high-confidence quantification down to the part per million or even part per billion level. Having that granular data is your ultimate compliance benefit. It gives the FDA irrefutable proof that your product remains safely below the acceptable intake limit. Acquiring that proof is heavily capital-intensive. You have to tie up mass spectrometers that cost hundreds of thousands of dollars and dedicate specialized analytical chemists to run them. LC-MSMS demands the highest investment of time and money your lab can make. Yet, it remains the only analytical tool capable of definitively clearing a high-risk drug for the market. Once you complete your risk assessment and run your tests, Step 3 of the FDA mandate requires establishing a permanent control strategy over the lifecycle of the drug. Effective control requires aligning your testing costs with the specific chemical vulnerability of the drug product. This quadrant chart maps the drug's complexity against the mitigation investment required. If you manufacture simple APIs that lack secondary amines, your strategy stays strictly on paper. You invest in a robust risk assessment to prove nitrosamine formation is chemically impossible, omitting physical testing entirely. If you have a stable legacy formulation with known precursors, you implement HPLC limit testing. It provides a routine, cost-effective safety net for batch releases. But if you are dealing with complex nitrosamine drug substance-related impurities, you bypass the lower tiers. You immediately adopt full LC-MSMS quantitative procedures to ensure precise control. Correctly mapping your product to this matrix protects your operations. It prevents regulatory failures on high-risk drugs and stops you from draining your laboratory budget on unnecessary testing for low-risk ones. The regulatory mandate on nitrosamine impurities is permanent, and global health authorities are strictly enforcing it. The costs of running these risk assessments and mass spec analyses are daunting. Treat them as a mandatory business investment required to defend your pipeline's profitability. If you manage a low-risk portfolio, your objective is to front-load your chemistry defense on paper. Build comprehensive risk assessments that legally protect your laboratory resources for the projects that truly need them. If your pipeline relies on complex formulations or vulnerable amines, the time for cutting corners is over. Secure the budget for quantitative mass spectrometry now, before a batch fails. The multi-million-dollar cost of upgrading your analytical testing is a steep price to pay, but it is only a fraction of the devastating financial and clinical cost of an FDA-mandated global recall.